Microbiologist Julie Huber, Plankton Ecologist HEIDI SOSIK, and Biological Oceanographer Nina Yang. Stay with us. Marine Microbiologist Julie Huber, Plankton Ecologist HEIDI. Hi everybody. In just a few minutes we'll get started with Woods Hole Oceanographic Institution's Ocean Encounters presentation, Small but Mighty, Microscopic Marine Life and its Outsized Role in the Ocean. We've still got a lot of people joining us, so thank you for your patience. Hello, you've joined Small but Mighty, a Woods Hole Oceanographic Institution Ocean Encounters presentation. We have a lot of people joining us tonight, so please bear with us. We'll get started very shortly. And now, please take a moment to read WHOI's land acknowledgement.
The ocean covers 70% of the globe. It gives us oxygen and food and millions of jobs. It brings joy and shapes our climate and weather. The ocean is life, and it belongs to everyone. Woods Hole Oceanographic Institution is the world's independent leader in ocean discovery, exploration, and education, working to understand and sustain one of humanity's most precious common resources. Join us today for our ocean, our planet, and our future.
Welcome to Ocean Encounters, a series from Woods Hole Oceanographic Institution, or WHOI as we like to call it for short. Tonight's event is Small but Mighty, Microscopic Marine Life and its Outsized Role in the Ocean. My name is VERONIQUE LACAPRA. I'm the host of Ocean Encounters. We're now in our 6th season, and I'm happy to share that our series has once again been named an endorsed activity of the UN Decade of Ocean Science for Sustainable Development. I'd also like to thank our sponsors. WHOI's Ocean Encounters events are made possible in part by the Avatar Alliance Foundation and Dalio Philanthropies. Thank you to them. Thank you also to any WHOI members who are joining us tonight. You are the heroes at the heart of our efforts. Before I introduce our speakers, I want to let those of you joining us on Zoom know how they can participate in tonight's live conversation. If you have a question for one of our panelists, please use the Q&A button on your Zoom screen and type your question in the little window that appears. You might be more familiar with the chat function in Zoom, but please ask your questions using the Q&A button instead. We often get hundreds of questions, so I apologize if we don't get to yours while we're live. You can ask any questions anytime, starting now. I also want to let you know that we are recording this event, and that recording will be made available on the whoi.edu website. We had more than 1,670 people pre-register to join us tonight, so you're in good company. We also have viewers joining us right now on Facebook, YouTube, X, and LinkedIn. Thank you and welcome to everyone. Let's get started. Microbes may be small, but they have a big part to play in shaping our planet. The ocean's smallest inhabitants form the base of marine food webs, help regulate global climate, and may hold the secrets to the origins of life on Earth. Here to help us explore invisible ocean life and its profound and far-reaching impacts are 3 WHOI scientists. They are marine microbiologist Julie Huber, plankton ecologist HEIDI SOSIK, and biological oceanographer Nina Yang. Welcome, everyone. Let's start by having you each introduce yourself. Tell us your area of expertise and very briefly what you work on. We'll get into more detail about all of that throughout the evening. Let's start with HEIDI, then Julie, then Nina.
Hello, everyone. My name is HEIDI SOSIK. I'm a senior scientist at WHOI. I have been involved in research on microscopic life in the ocean for more than 35 years. One thing I spend a lot of time on these days is leading long-term ecological research. We have a large interdisciplinary, multi-institutional effort to understand how planktonic ecosystems are responding to ocean changes across the continental shelf off the northeastern US.
Wait, tell us what plankton is.
What plankton are?
You said planktonic, yeah.
Plankton are tiny organisms, photosynthetic organisms, and little animals that free swim, float in the ocean, and they live everywhere. And you're going to hear a lot more about them tonight. They're really amazing creatures.
There you go. All right, Julie.
Hi, everyone. I study life in the dark at the bottom of the ocean, and I think about how life's smallest organisms, or microbes, can harness chemical energy, not using the sun, but other sources of chemical energy in the ocean to support themselves and some really remarkable ecosystems in the deep. So my work takes me far away from Cape Cod, where we live, to some really deep and remote places. I get to use some cool remote technology, deep submergence technology, and kind of a toolbox from biology and chemistry to study these systems. And I'm also interested in some of the things we're going to talk about today. How can we use what we're learning from this basic research to inform some big concepts or questions and problems, such as conservation, resource extraction from the ocean, solving the climate crisis, and as you already mentioned, Veronique, even the search for life beyond Earth.
All right, and Nina.
Hi, everyone. I'm relatively recent to the HEIDI community. I'm a postdoctoral investigator here, and I've been with HEIDI for about a year and a half. I have the opportunity to work across both the biology department with scientists Annette Govindarajan, as well as the Marine Policy Center at the institution with DGen. And here, my interests are really on how do different organisms, from really small microbes all the way up to huge whales, interact in the ocean, and how these interactions affect the ocean ecosystem. And also through the policy side, trying to use this research to help inform better decision-making to protect our ecosystem and the benefits. And I'm excited to be here with HEIDI and Julie and to talk a little bit more about this today.
All right, awesome. Julie, I want to go back to you to start with. Help us understand exactly what we're talking about tonight. So HEIDI started to talk about plankton, but what are ocean microbes? What do they include, and why are they important?
So microbes are single-celled organisms. We're multicellular organisms. This includes phytoplankton, protists, bacteria, and archaea. And they make up the vast majority of ocean biomass. And as we'll be learning, they form the base of food webs. And they've had billions of years to evolve on our planet. So they are absolutely everywhere. But we mostly hear about microbes sort of in negative association with our health, whether it's an ear infection or that lettuce that you shouldn't be able to eat. But they do all these amazing things on our planet. So they're a really important source of food, for example, and important drugs in the fight against disease. And in the ocean, they're really critical for the health of the sea. And microbes are just amazing chemists. So they're capable of harvesting energy from almost any chemical reaction, and they use that energy to grow and thrive. So they're responsible for about half of the oxygen on our planet produced by the types of organisms that HEIDI studies. But they also form these really neat symbiotic relationships with many animals in the ocean. So this includes these really beautiful deep sea tube worms that we find at hydrothermal vents, but also in many other animals, such as corals, where they are providing essential nutrients to the host. They even help us clean up our own messes, such as this image from the Deepwater Horizon oil spill, where some of the first responders to this event were actually bacteria living in the ocean who could degrade components of that spill. So whether we're in the surface ocean, the deep ocean, microbes are transforming things like carbon and nitrogen and other elements. They're a key component of ocean biogeochemistry. And as we'll learn from Nina, they really serve as the base of the food web throughout our ocean.
So I mentioned, and then you mentioned again, how microbes might somehow be connected to the origins of life on Earth and maybe even beyond Earth. Tell us about that.
Sure. Well, the first evidence, our planet's about 4.5 billion years old, and we have really good evidence of life about 3.7 billion years ago. And for most of Earth's history, that life was microbial. And so our last universal common ancestor was a microbe, that everything else came from that. And so by studying some of these ancient ways of gaining energy, we can potentially learn about how life originated here.
And what about other places?
Well, Earth is not alone in having an ocean within our solar system. So we now know there are a number of moons of Jupiter and Saturn, for example, that have oceans under an icy shell. And so the lessons we're taking from how life can thrive in the bottom of the ocean, far away from the sun, are lessons we can now apply to the search for life beyond.
Pretty cool. Okay. Heidi, we're going to zero in with you on a subset of the microbes Julie was talking about, the phytoplankton you mentioned earlier, also known as algae. I guess my first question is, are all algae single-celled? And then why should we care about these tiny little plant-like things that live in the surface ocean? How do they affect us?
Yeah, well, they affect the whole planet. So first, definitely there are macroalgae. People will be familiar from the beach with seeing seaweed and kelp and macroalgae that live in that special environment near shore. But it turns out that most of the algae are single-celled, they're microbes, and they are much more abundant and everywhere in the ocean where the sunlight is shining. So these phytoplankton, as they're called, these free drifting single cells, they have this incredible special power that they can photosynthesize. And what that means is that just like plants on land, they make special molecules like chlorophyll and other pigments that they can use to capture energy from sunlight. And then they can use that energy to grow and reproduce. So unlike us and other animals, they don't need to eat. They just capture sunlight and photosynthesize, and voila, they can grow. Unlike plants on land, which we can see by looking out the window, these phytoplankton are so small that we need microscopes to see them. But when we do zoom down, we see, at their scale, that they're incredibly beautiful and diverse. You see some examples here. And they come in all kinds of sizes and shapes. The tiniest phytoplankton are so small that you could line up 100 or more across the width of a single human hair. And then the largest species that you're seeing here, they can stay together after they reproduce. So the single cells remain in larger colonies or chains and survive in certain conditions in the ocean. And they're much, much bigger than the tiniest phytoplankton. As part of my research, we actually collect millions and millions of images like these. And I never get tired of looking at them because they're so interesting and beautiful, kind of like, nature's artwork, I think. And in fact, people do use them to inspire and create art. These tiny cells, though, they're much more than beautiful. They are huge players in ocean ecosystems, and they even help control the climate of the whole planet.
Now, Nina, you study everything from tiny microbes all the way up to things like whales and sharks and other big marine life. So help us connect the micro to the macro.
Yeah, I think, so I come from a microbial background. In my PhD, all I did was study microorganisms, and I sort of ignored everything bigger than that. But coming into HEIDI, the work that I do here, everything in the ocean is sharing living space. So to only think about one particular group of microorganisms, for me, it just wasn't a complete picture. And so everything is connected, they're living together, and they're often interacting in the environment. And so a certain type of interaction we can think of are food web interactions. And that's basically just talking about who eats who, right? And on land, we have this example. We have plants, and you might think of cute little animals like hamsters or mice eating these plants. And then these mice get eaten by larger animals like foxes. Cows also eat plants, and we eat cows. And so that's sort of a food web there on land. In the ocean, we have similar dynamics like this. Heidi already talked about photosynthetic, these phytoplankton, and they are often considered the base of the marine food web. And we have larger microorganisms like ciliates that are predators, and they eat these other microorganisms, including phytoplankton. And then these microbes are then fed on by smaller animals like krill, which are also being fed on by slightly larger animals like jellyfish or smaller fish. And then we have the really big charismatic megafauna that you might all be familiar with, including my favorite, like whale sharks, that will feed on some of these smaller organisms. And then, of course, you have large apex or top predators that can eat a variety of animals that live in the ocean. And so food web dynamics and these interactions from the very small to the very large are more than just who's eating what, but they represent this flow of energy and also carbon in the ocean. And what's really cool is that they're all fueled by these little microorganisms.
So you touched on how these food webs play into energy and carbon. I want to kind of flip that around, Heidi, and ask, how do changes in carbon in the atmosphere and climate change in general affect microscopic marine life? And that's probably a pretty big, complex question, but maybe just give us a sense of the answer. What do we know? What we don't know?
Yeah. So for sure, there are many kinds of climate change impacts in the ocean. And then, of course, there's also big feedbacks on climate from the ocean. One of the most important examples of climate change effects is that the ocean has been getting warmer and warmer. You see from this map here, the really bright reds are areas where the ocean is now warmer than it ever was before. And as these water temperatures rise, it's affecting things really in complicated ways, like the layering of the ocean, what we call stratification. When you have warmer waters, they're lighter and they tend to sit on top of the colder, deeper layers of the ocean, kind of capping them off or isolating them. And that might not sound like it's very important for plankton, but it turns out it really is. For phytoplankton, they need to be in the upper ocean where the sunlight is shining so they can photosynthesize. But the deep cold waters are where most of the nutrients in that, like nitrogen and phosphorus, that the phytoplankton need to grow, those nutrients are isolated in those cold, deep waters. It's the same kind of nutrients that you might, fertilize your houseplant or fertilize your garden with. So when you have this strong layering in the warming ocean, you're kind of isolating the nutrients and keeping them away from where the phytoplankton need to grow. So
that warm water just sort of acts like a cap on the cold water.
Yeah. So in some places, that warming impact, it may be actually lowering ocean productivity and even shifting the kinds of phytoplankton that we find. The smaller sized cells have a kind of nutrient uptake advantage, so they might be able to outcompete some of those big chains and colonies that you saw earlier as the ocean gets warmer and warmer.
Interesting. Nina, given what we know about ocean food webs, can you comment on how climate-driven changes in microscopic life might be playing out at higher trophic levels, fish, or even us, since we eat seafood?
Yeah, I think that's one of the many reasons why it's been so important to do this kind of work, to understand, what's happening in the ocean and how it impacts the ecosystem. And so actually a really relevant example is from algae. So you may be familiar with harmful algal blooms in the surface ocean. And in some regions of the world, the ocean warming that Heidi just talked about is causing an increase in these blooms. And harmful algae, and you can see this image here, they're caused by microscopic organisms that under certain conditions can trigger super fast growth, and they basically just explode and sort of take over that system. One example of a harmful algal bloom is Alexandrium, which is what's causing the image that you're seeing right now. The reason why they're harmful is because not only do they bloom and grow a lot, they also produce a toxin that can cascade through the ecosystem and the food web and make animals and people sick. And so you may be familiar with shellfish, like mollusks or scallops and mussels. They filter microorganisms in the water when they eat. And when you suddenly have all these harmful algae in the water, they end up eating that harmful algae, storing the toxins in their body. And when we eat shellfish, we can get shellfish poisoning, which can lead to death in severe cases. And so there is a lot of active effort to monitor these blooms so that we can limit our exposure to toxins. But one of the ways that we do this to control our exposure is actually just to shut down the fisheries, and until the blooms subside and the toxin levels go back down. And that's harmful to local economies that would depend on these fisheries. And so it's one of the examples where you have small microorganisms that can really have a huge impact in the food web. And in general, I think I want to emphasize that these food web interactions are really complex and actually quite difficult to predict how climate change at the bottom of the food web is going to cascade up to higher trophic levels. But this is one example that shows that there are big impacts to larger organisms and to people that research is really important for to better understand.
So Julie, if HABs are an example of changes in microscopic life in the surface ocean and along the coast, are there human impacts that are reaching all the way to the deep ocean and the microbes you study there?
Yeah, well, I really liked what Nina said about how everything's connected, right? And it's important to remember that it is one ocean on our planet, right? So what's happening in the surface ocean or along our coastlines, it eventually impacts the deep sea. And, as an example, I've seen lots of human trash at, 16,000 feet in our ocean. But one really important concept to think about in the deep sea is just how it is a huge, giant reservoir of carbon on our planet. So this export of carbon from the surface to the deep takes place through things like what you're seeing in the video now, through this passive sinking of particles. This is what we call marine snow. And I've seen this myself traveling down in the submarine Alvin to the seafloor. It's really cool to see this kind of rain down of material. And that's basically any carbon and leftover stuff that wasn't consumed. It eventually makes its way to the deep sea where it's captured in rocks and sediments, and it's kept out of the atmosphere for a really long time. So your question then was about these human impacts affecting the deep ocean. Well, given that the deep ocean is such an important reservoir of locking up carbon and keeping it out of the atmosphere, almost any human activity that impacts the deep ocean could disrupt this process. So, in here I'm showing that, the deep ocean is really the largest reservoir of carbon on our planet. That GT means billions of tons of carbon. So there are new and emergent uses of the deep sea that could really threaten this process of carbon storage, but also the life there. This includes things like deep sea mining or changing the carbon budget. And so I really think there's this urgent need for more studies of these environments, because I can't even tell you if microbial life has been impacted by the climate change, for example, because we have so few sort of baseline studies of how life works down there.
Yeah. All right, I want to bring in our audience. Let's go ahead and take our first audience question. We have my colleague Ken Costell in the background fielding those questions. Do you have something for us, Ken?
I do. There have been a relatively low number, but they've been very good. We could have asked almost any of them. Thankfully, you guys already answered quite a few. However, Ethan is a sophomore here at Falmouth High School. And he's wondering, since you're talking about impacts, whether you have seen any impacts from microplastics on marine microorganisms, particularly the toxins that are contained in plastics, either in the surface waters or for Julie down deep.
Julie, you want to answer this, I can tell.
No, I'm not sure I want to answer it. Well, one thing I would say, Ethan, it's a great question. There are microbes that can actually degrade components of that plastic. So there's kind of emerging research that shows there are certain microbes that can colonize plastics and degrade certain components of it. And there's actually a group of researchers here at WHOI who are trying to figure out that mechanism to make biodegradable plastics. I'm going to defer to Nina on this, because I think where we're seeing the bigger impact of microplastics in the ocean is that cascade up in the food chain.
Yeah, I'll just briefly touch on it. Microplastics, I think is like one of the hottest issues right now. And there's been a lot of research over the years of finding plastics in animals, and to some case where their stomachs are full of plastic, they're not able to eat other foods. And so we know, at least from perhaps accidentally replacing their desired food source, whether it's plastic bags that look like jellyfish or other pieces of plastic that can look like prey items for some of these animals, it is affecting those higher trophic levels. Something sort of like a pressing issue right now that we're all trying to do more work to understand.
Yeah,
There
I
are
think
studies that
Sorry,
that
go ahead.
have shown the microplastics in plankton as well, but for sure we need more research to figure out what the impacts of that is. But they're there.
Yeah, I think we know more about sort of those physical kinds of impacts that you were talking about, Nina. An organism with a full stomach or a plastic bag in their system, but but still to be figured out is what's happening to the chemicals involved in plastic, right? Let's take one more if you have one, Ken.
Okay, I'm going to go over to YouTube for one we got from Biology in a Nutshell. What are your favorite plankton types? Or Julie, favorite microorganism in the deep sea?
So my favorite microbe is this group of microbes that's ubiquitous on our planet. They're called methanogens. They make methane, which is a really potent greenhouse gas. But they live, there's a lot of them in the deep ocean. And what I like about them is their metabolism is super simple. They take CO2 and they take hydrogen and they make methane. And they can grow from super, super cold temperatures up to like 120 degrees Celsius, which is, if I'm doing my math right,
really hot,
280 degrees Fahrenheit. So they're just this remarkably successful group of organisms. They live in the gut of cows, they live in rice paddies, and at the bottom of the ocean.
Heidi, your favorite phytoplankton?
I don't know if I can pick just one. I might have to talk about two. So on the very small end, these phytoplankton called picocyanobacteria, they are almost ubiquitous in the surface ocean. They're so abundant. There's thousands and thousands and thousands of them in a spoonful of surface seawater. And they've been on our planet longer than just about any other organisms. They are actually were originally responsible for the oxygen in our atmosphere. So they're really, shapers of our planet. And then on the other end, there are these diatoms, which are incredibly beautiful. Some of the most intricate shapes that you saw earlier in the presentation were diatoms that they make these glass shells that are really beautiful, and they're extremely successful in the modern day ocean and important for some of the food web things that Nina was talking about. They're really good for fish.
And they're on the heavier side, right? So they might be responsible for a lot of the carbon that's actually making it further down into the deep ocean.
Yeah, because they have those glass shells. If they die or they're damaged by some grazer, they actually sink very fast because they are heavy.
All right, I want to get into now how each of you actually does your research. Nina, you use a kind of forensics. You're a little bit like an ocean detective. Talk about that for us.
Yeah, I really like that title. I probably should have replaced biological oceanographer with ocean detective. I work with something called environmental DNA, or eDNA for short, is probably what I'll be talking about, how I'll refer to it. In its simplest definition, it's just all the DNA or genetic material you can find in a given environment. And in the ocean, it can take on many forms. So we've been talking a lot about microorganisms. DNA in microorganisms can be these single cells because they're small and we can collect a lot of them. For larger animals, you can't really collect a whole fish and take all the DNA and call it a day. We're actually collecting pieces of these other animals. You can think of maybe they're shedding skin cells or scales, or their poop. If they're pooping and the poop stinks and we just happen to catch it, there is DNA there. And so this ocean detective thing that I love is that it's really difficult to study an environment like this, like our global ocean. It's huge, it's deep, it can be really dark and really cold. And DNA, and being able to use eDNA gives us this evidence that the animals or other microorganisms are there in the water. So they, as organisms live in the water, as they swim and move, they leave behind traces that they were there. And scientists can collect the water, drain out all the water, and just keep the material that we have left on filters. And then we can use those filters to extract the DNA from there. And with that DNA, we can figure out who that DNA belongs to, so who's living in the water. And also with enough information, we can start to tease apart their different behaviors. Who are they sharing space with? What are they doing? And so one of the things that I think has been really cool working at HEIDI, I talked about biologist Annette Govindarajan, and we work with robots that can collect water autonomously. You're seeing some clips of me doing field work where we're prepping these robots and these samplers, because we need a lot of water to be able to study an ocean of this size. And these robots, we put them into the water and they just pump and we can bring them up and have filters ready for it. Otherwise, traditionally, we use large bottles that we haul out of the water, and then we collect all the water into smaller bottles, and then we do our filtering. So being able to work with these different robots and developing technologies, in addition to this eDNA sort of forensics work, is really pushing us forward to be able to understand what lives in the ocean from very small to the very large.
Okay, so that's kind of all happening nearer the surface. Julie, how are you finding out about microbes way down deep at the bottom of the seafloor, really, and even below the seafloor?
Yeah, well, I think it's awesome that we all study these tiny little things, but we get to use these really sophisticated robots to get our samples often. So I use a lot of different deep submergence vehicles. This video is one that WHOI operates. This is the remotely operated vehicle, Jason. And this was from a cruise I led last summer. We're working at a deep sea hydrothermal vent called Axial Seamount off the coast of Oregon in the Pacific Ocean. All that purple and white stuff that you're seeing, most of that is microbial. So with these remotely operated vehicles, there's actually no people in them. The vehicle is tethered back to the ship where all the scientists and engineers are sitting. We have like 20 different screens where we can see beautiful images like this. This is filamentous bacteria. And on this expedition, we were studying the microbes in these venting fluids. We were interested in how fast they were being consumed by single-cell protists. So the advantage of using this type of technology is that the vehicle faces the harsh conditions while we're all safe on board, and we kind of trick out the vehicle with all these fancy sampling devices. You can think of them as like the really fancy and expensive vacuum cleaners to suck up water. Some of them are made of titanium. And we have even developed technology here at WHOI so that we can actually do the experiment on the seafloor and not even have to bring the samples back.
So, and Jason is remotely operated, right? So there's kind of a control center on the ship.
That's
from
right.
Which Jason's being controlled.
Yeah, command central.
Yeah, exactly. I have sort of a weird question that just occurred to me. I've seen those beautiful purple microbes now several times, but why do you need to be purple if you're at the bottom of the ocean and it's all dark?
Yeah, we don't know. And so this is a group of ciliates that's found in a lot of different places, and it always has this pigment. There's been 1 or 2 studies showing there are some like bacterial symbionts living in association with that protistan ciliate, but we collected a lot of it, and I'm working with some folks to try to figure out what is going on. But we see it at a lot of the hydrothermal vents in the Northeast Pacific Ocean, which is where this footage was taken from.
That's so beautiful. So can you actually bring microbes up to the surface that normally exists at the bottom without them? I don't know, because the pressure is so different. How do you handle that?
Yeah, well, there's a lot of environmental changes, right? Pressure, temperature, energy. So there are some microbes that grow fine at the bottom of the ocean. They also grow fine here in my lab. The pressure change doesn't seem to bother them. But there are others that will not grow in my lab unless I impose high pressure in the laboratory on them. Those are called barophiles. So they need that high pressure to grow. And similarly with a lot of the organisms in this environment, they will not grow at room temperature, right? I have to put them in an oven in my lab to get them to grow. So we do the best we can to recreate that environment in the lab.
Oh, okay, cool. HEIDI, phytoplankton, they're at least near the surface, but as you were saying, there's so many different kinds, so many of them. What tools do you have available to be able to make sense of all that diversity and figure out what they're doing?
Yeah, right. So I love collaborating with Nina and her colleagues because the eDNA approaches are really powerful. But of course, another way to see that great diversity is to look through a microscope. And marine ecologists have been doing that for decades and decades. That traditional way of doing that work, though, is just too slow and too time consuming. We
I did
always
some of
find
that
that
in graduate school with freshwater. I know what it's like.
yeah. And, to study the big ocean and how fast it's changing, we just need more and more observations so we can understand those changes and see them. And so for that reason, my colleagues and I at WHOI, we worked on inventing new technologies. You see here a robotic underwater microscope. Back when we were developing it, we call it Imaging Flow Cytobot. This instrument can go underwater, and it's a robot. It can take pictures of thousands and thousands of microscopic plankton every hour, and it can do that by itself underwater hour after hour for many months. While, in fact, there's one of these in the ocean off Martha's Vineyard. While I'm talking to you, it's taking images of what plankton are there, and you can actually see that on our website dashboard. This kind of robotic observation of microscopic organisms is giving us huge data sets. We now have billions of these images, and we're needing to analyze them with AI to help us know what species are there, how their abundance is changing over time. And we're doing that both at ocean observatories, but we also take these automated instruments on our research ships, similar to the cruises like Nina was talking about. We go out and use these automated imaging technologies to map patterns of change in species from place to place. In our long-term research, we go out every season, year after year, so that we can do this kind of mapping through space and time. And then
there we're seeing you use a tow sled, right, called Stingray, that just gets towed behind the ship, but it has one of those imaging systems on it.
Yeah, exactly. So this on this what you're seeing here is this tow sled being deployed behind the ship, that it can fly up and down through the water column, and it's actually imaging larger creatures that eat phytoplankton. So as soon as we start studying phytoplankton, we immediately have to figure out, well, who's eating them and what is the impact of them on the rest of the ecosystem. So we use multiple kinds of imaging to be able to look at plankton across a big part of the food web. We also use both the observatories and the ship-based observations. We put those in the context of even larger scales of observation of the oceans that we can do by connecting the plankton observations we're making in the ocean to the kinds of plankton observations that we can get from Earth orbiting satellites looking down on the ocean. So in that way, we can look across, not just the place where our observatory is or where our ship is going, but try to scale up to the whole ocean basin and even global scale impacts of these tiny plankton.
Wait, but if phytoplankton are so small that you can't see them without a microscope, at least some of them, how can satellites see them from outer space? How does that work?
Yeah, it's super cool, right? How it works. If you look down from above on a forest or a meadow or a cropland, if you've seen pictures from aircraft or looked out the window as you're flying over, you see all of these different shades of green, right? Especially in the spring and summer when all of those organisms are growing. That color that you see when you look down, that's from those chlorophyll and those other pigments I was talking about that they use to photosynthesize. And remember, phytoplankton have those same pigments. And those pigments, they actually shift the color of the light that's scattered out of the ocean, just the way leaves change the color of the sunlight that scatters back to your eye. So they're really tiny, but there's so many of them that that places in the ocean that look darker and greener are because of those plankton. That's where there's more plankton pigments in the water. What you're seeing here is this amazing satellite-based view of our global biosphere that's produced by NASA. You see a vegetation index on land, and then chlorophyll concentration in the ocean. So the places where you see those deep blue colors, there's not very many phytoplankton pigments in the water. And then the places and times where you see the bright reds and yellows, there's a lot of plankton in the water. So we see these amazing patterns of change from this kind of space observation. So
Yeah, it's
cool.
a great animation. I love that. Let's take some more audience questions. Ken, what do you have?
This, I think, is for Nina. Joe asks, how long does eDNA persist in the environment?
That's an excellent question. And I'm going to say it's unknown, because there's a couple of different factors. So sometimes we call this study the ecology of eDNA, where we need to know who's shedding eDNA, how much they shed, and how long different types of eDNA persist and what kind of environmental factors can influence their persistence or movement or degradation in the water column. So you can think about it in the surface ocean, it's very, very warm, but down in the deep sea, in the regions where Julie goes, it can be very, very cold. And these different conditions can impact that persistence. And so I wish I could say an answer like a couple of hours to a couple of days, but it's a really exciting unknown field that we're still trying to figure out.
It's kind of like putting the
Joe-
eDNA in the refrigerator when you're down
Yeah.
where Julie's
And
working.
then we actually know that different animals shed DNA differently. Like maybe a fish will shed more than, for example, a jellyfish. So there's a lot of unknown. The application of eDNA for animals is quite new, whereas we've been using molecular techniques like this for microbes for quite some time. And so there's still a lot of things that we need to figure out.
Yeah, I was also just going to add from the microbial perspective, there are microbes in the ocean, like in deep sediments, for example, they can lie dormant for, we think, thousands of years, just barely maintaining the integrity of their cell, but not dividing. And we can detect their DNA. And I think it's why a lot of scientists like Nina and Heidi, we aren't only using DNA, right? We're using other tools also to convince ourselves that they are active or viable, or they're moving, or they're doing these important processes in the ocean.
We can take another question, Ken.
Okay. Somebody else asked if you can test samples remotely without bringing them to the surface, might this be possible in the future on ocean moons of Jupiter and Saturn?
Julie.
Yeah, that's a great question. I will say that most of we call it in situ science, so in the environment science that we've been doing with microbes is we're basically running the experiment on the seafloor and the experiment stops, and then we bring it back to the lab and analyze it. There is one technology that is used here at WHOI to detect harmful algal blooms in real time, and that is a DNA-based technology where basically the instrument can bring in seawater, can extract the DNA, and probe for certain organisms that are indicators of a harmful algal bloom. And that has been deployed once in the deep ocean, but it's still pretty rare to do it all the way to the end of the experiment in the ocean. So when we think about a spacecraft going to the outer solar system, we can't only do biology, right? The geochemistry of our planet is, intricate and so important to how biology functions here. So for these missions, we're going to be looking at all sorts of biochemistry, chemical indicators, as well as some biological indicators. There is a dream to sequence DNA in space, but it hasn't been realized. And I personally would love to do it at the bottom of the ocean before we take it, on a 7-year flight to the outer solar system.
Approaches like imaging microbes can also be automated. The imaging flow cytobot also is used to detect HABs, and there's no need to bring a sample back. You just send the image data or the machine learning result that yes, there's a HAB alert in the water where the instrument is deployed. And in principle, that kind of imaging could be done in the deep sea and on other planets. It's a technological challenge for sure, but the robotic techniques do exist, and we use them in the ocean.
Yeah.
I want to shift gears a little bit and talk about your careers. You're all women in a field, oceanography, where women have historically been underrepresented. In fact, Julie reminded me that in the early days of WHOI, women were not even allowed on research ships. And now all of you are seagoing scientists. I'd like to hear from each of you what got you interested in oceanography in the first place, and then sort of what's motivated you to stick with it. Heidi, let's start with you.
Yeah, so I think for me, the stage was set really early. I grew up spending a lot of time in the outdoors and in a family that really encouraged me to appreciate so many things about the natural world. And you see me here with my dad, really to embrace that natural world with curiosity and real joy. I definitely, as a young kid, I wasn't thinking about being an oceanographer at all, though that wasn't until college. I I encountered an advisor who was an incredibly great role model and helped me discover the path to oceanography. And that advisor, her name is Penny Chisholm, she continues to be an incredible mentor and colleague to me so many decades later. So the value of these kinds of relationships and are shaping careers like mine are so powerful. You see this picture of us here together just a few years ago at the TED conference, where we were kind of supporting each other through all the nerves of we were both giving TED Talks about life in the ocean. And it was incredible to be there with her and to have spent all these decades with her as a mentor and colleague. And
Yeah,
I just want to, yeah. For me, all through my career, this connection of doing research with wonderful groups of people just continues to be inspiring. I just got back earlier this month. You see us here on one of our seasonal plankton research cruises, and I was leading this team of scientists. These are mostly early career scientists, a different group of people we go out with every time. And they're just, approaching the research with such dedication and diverse perspectives and that same kind of curiosity and joy of discovery that I felt so much as a kid. It just makes it so fun and rewarding to go to work every day.
Julie, how about you?
Yeah, well, for reasons I can't really explain, I've always been just kind of enamored by the ocean. I grew up in the Midwest of the United States, so I grew up outside Chicago and Ohio. No one in my family was a scientist or an engineer, but I am part of the Cousteau generation. I even share a birthday with Jacques Cousteau. But I really kind of credit books, TV shows, aquariums for kind of sparking that fascination. But honestly, it was my parents who, despite being English and history buffs, they let me pursue this passion. We went to the ocean or the Great Lakes. I learned to boogie board in the Great Lakes to kind of pursue it, and they helped me find that path forward in college. So once at college, throughout my schooling, I also had really wonderful mentors. Unlike HEIDI, they were all men, but they gave me so many amazing opportunities and advice along the way. They still do today. So I know I had a lot of privilege in that path. And so one of the things like HEIDI that I work really hard at now is trying to pay that forward and give the opportunity to others. The ocean, as we're talking about, is facing a lot of challenges, but there's also these just completely amazing stories to tell. So the ocean needs more eyes on it, and we need that larger network of diverse individuals across the globe, right? Really working in science, in engineering, in research and policy translation. We need an educated voting public. And so I stick with it for a lot of reasons. Mostly I care really deeply about the ocean, but like HEIDI, I really love working collaboratively with others to help study it. This is not a science where you're, by yourself in a lab for hours on end. And by working with these groups of people, we can really enable, especially early in their career, their own path of scientific discovery.
Julie mentioned policy. I know that's been important to you, but tell us a little bit about your career so far.
Yeah, so it's actually been really cool to hear what Julie and HEIDI have to say, because I can see bits and pieces of their story sort of resonating in my own trajectory. I think mentorship has been really important to get me in and also to keep me in, but also support from a network and exposure to different things. But I would say I grew up, that's a picture of me in a rainforest, which is not the ocean, but I'll touch on it in a second. But I grew up in New York City, which is not really well known for nature. It's really more well known for large buildings. And my parents are also immigrants who didn't really speak English or didn't really know what education was like in the US. And I like to share that because they sacrificed a lot for me to have more opportunities. So I convinced myself for most of my life that if I wanted to make money and have a job, and I wanted to do something in science, I had to be a medical doctor. And so I went all my entire life very convinced, all the activities that I did, that I was going to be a medical doctor instead of an ocean doctor. But in college, I went to women's college, I went to Barnard College, and I had such great mentors and exposure to different types of science, and in particular, microbial ecology. And so I had an opportunity to do field work led by a woman scientist, Dr. Krista McGuire. She's up on top. It was a team with other female college students, and we lived together in the rainforest, climbed hills, and collected soil samples, and I got to do my senior thesis on it. And it really was not only my first exposure to microbial ecology research, but also an exposure to the idea that I didn't have to be a medical doctor to do science and to have a job. I could really do anything that I wanted. I think what brought me into a PhD in oceanography, and a lot of the work that I've done is focused around climate change impacts, is because I sort of grew up in this generation where we had a lot more awareness, I think, of that climate change was happening. And I felt like I was getting more and more anxious that I needed to do something to make an impact. And the year that I applied for grad school in 2016 happened to be a record-breaking year for surface temperatures. And throughout my PhD, I wanted to focus on what those impacts would be on microbes in the ocean function. And I also had this opportunity to actually get engaged in policy. As a Canals Marine Policy Fellowship, which is through NOAA here in the US, you can work in Washington, D.C. for a year. And I got to work with a wonderful team of people who put together the first ever US Ocean Action Climate Plan, which is a document that outlines this roadmap to develop and implement solutions on how we can harness the power of the ocean to tackle the climate crisis. And so that was a really awesome opportunity, and it really brought me here to HEIDI to bridge that, continue bridging that, to do biology and policy.
Yeah, so talk a little more about that. So how does the work you're doing with Annette Govindarajan on eDNA connect to policy?
Yeah, one of the things that I learned as in government is that knowledge is power. So if you have a lot of information, you can actually use it to make good decisions. But if you have no information, you can't really do much, or you make decisions that might not help the environment or human society. And what I learned is that there's not a lot of data for life in the ocean. Even though there's quite, we have a lot of research, and I spent years studying it, there's huge pockets of the ocean where we've never gone to, or we've never sampled. This is just a quick schematic to show you where we have data on life in the ocean for the mesopelagic or the ocean twilight zone, which is right below the surface, not quite as deep as hydrothermal vents, but in the midwater. And everywhere where you see white, that is where we don't have any data. Even where there are blue dots, that's actually like one data point since the year 1900. So we just don't have enough information to make the necessary decisions to protect this ecosystem, which also provides us with so many benefits. So this is where I think eDNA is really exciting, because it's a scalable tool that we can apply to collect all this water and quickly figure out different kinds of organisms from one sample. And so hopefully we can be able to develop these technologies even more to be able to develop a baseline understanding of life in the ocean.
All right. We're running out of time a little bit, but I want to take at least one more audience question. Ken?
Okay. Several people have pointed out that there are efforts to do things like seed the ocean with iron to stimulate phytoplankton blooms or stimulate upwelling, or even, somebody says, rightly, simulate the effect that whale poop has in fertilizing parts of the ocean. What are your thoughts in leveraging some of the processes that microbes provide for us naturally to help us in our climate crisis?
I think that one's for HEIDI.
Okay, well, I'll get started, but you all can help me. I think, when I hear questions like that, the first thing that comes to my mind is to emphasize that to course correct and to have the impact that we need to have as humanity on the trajectory of climate change, we need to reduce emissions. And that is like the first thing that needs to be done. The kinds of things that are talked about to try to manipulate the carbon storage systems in the ocean, I think, some of them certainly have the potential to help with mitigating climate on, the decades to hundreds of year time frame. But you've heard us talk tonight about a lot. Nina just emphasized how little we know about so many aspects of the ocean. And there's a real risk that if we tinker in systems that we don't, deeply understand, we could actually make things worse. The ocean is doing us an incredible service in helping to mitigate the climate impacts already. And in many of these topics, I think we should be very cautious about the fact that we don't necessarily have enough knowledge to know if the measures would increase or perhaps have other deleterious effects or even decrease the carbon services, the carbon storage services that the ocean is already providing.
Anybody want to add anything? Or are we good?
I think that's a great answer. I totally
Okay.
agree with Heidi.
So we are running out of time. I do want to ask you each one more closing question. Nina, you're just starting out your career, as we've talked about. What do you hope to achieve?
Yeah, so I want to start by saying that I've been following Julie and Heidi's research and science career for quite some time, as and so just to be able to share the virtual floor with them today has been a career milestone in my book. I really enjoyed learning from them. But so I think I'll echo some of the things that they said about what keeps them involved in science, and it's working with people. And I hope I continue to do really cool, exciting science and tackle the climate crisis. But what I really want to do is build a research program or some kind of legacy where it focuses on the people. And I think that we need to have scientists or people who are interested in science with diverse interests and backgrounds and experiences to be able to really address some of the greatest challenges of our time, and to also discover things that remain undiscovered. And that's where I hope I get to do in the future, is sort of develop this this legacy.
All right. Heidi and Julie, I'm going to ask you a slightly different question. What's been the most exciting discovery or experience of your career so far? Heidi, you go first.
Oh boy, that's a tough question, and I'd probably give you a different answer every time you ask me. There's so much we have still to learn, and every time we make measurements, we make new discoveries. But I think what I want to highlight right now is one example that's kind of in the front of my mind these days, that is about some new evidence that's making us rethink what we know about how phytoplankton die. We have these ideas that the classic food webs where the phytoplankton are eaten by the zooplankton that are larger than they are. The zooplankton come along and gobble them up. But recently we found in our image data that sometimes the phytoplankton are actually being killed by tiny parasites. The image you see on the top here is a healthy chain of diatom cells. The dark spots are where the pigments are that they're using to photosynthesize. And that cell's really healthy and happy. The one you see on the bottom image is the same species, but in this case, the diatom has been infected by parasites, and the dark areas are, some of them are actually parasite offspring. These little tiny parasites are able to get inside the single-celled phytoplankton, and they eat them from the inside out. And then to add insult to injury, the parasites actually reproduce inside the dead host, and then you see the little offspring of the parasites inside the diatom glass shell. They burst out and swim away to infect another cell in the population. So in that way, you can actually get like a parasite epidemic or outbreak that can kill a whole population and end a bloom. So this kind of discovery is just, it's really challenging our views of plankton food webs. We now think parasites are much more important than we previously thought. And some of our observations even suggest that they could become even more important as climate change continues to warm the ocean.
I have so many questions about that, but we don't have time. Julie, you get the last word. What's your favorite experience?
Well, my favorite thing about my job is watching people like Nina take over the world and succeed, which I think is just amazing. In terms of my own field work, I always keep kind of like a top hit list of amazing things that I've had the privilege to see at the bottom of the ocean. So like Heidi, the video that's about to show is really fresh in my mind because it was just captured this last December. I was working with an international group of scientists from Costa Rica in the US. We're working on Costa Rican seamounts. This footage was captured by a remotely operated vehicle called Sebastian, which is operated by the Schmidt Ocean Institute. This is at 6,500 feet deep, and you're watching these baby octopus hatch. So we had spent many weeks watching mother octopus brood their babies. And this is like brood their eggs, I should say. This is a very stressful process. It can take years, and the mothers are very stressed out. And, you know, to be in the control room, to be able to witness this happening, you know, there were people close to tears. It was just a remarkable privilege to watch it happen. And it was really exciting to then watch my Costa Rican colleagues kind of tell the story of this to their citizens. You know, 90% of Costa Rica is underwater. Most of that is in the deep ocean. And so, you know, kind of echoing what Heidi and Nina said, like every time we go into the ocean, we find something new, and we really need to be investing in people to kind of build this capacity, drive discovery, and to inform action in the global ocean. And, you know, it happens to also just be really, really adorable to see things like this.
Here it comes.
I could watch this all day.
I know.
Oh my
Be
God.
successful, young one, right?
Whee! That's so awesome. All right, we gotta wrap it up. I'm sorry. Unfortunately, that's all we have time for tonight. I want to say a really big thank you to Nina Yang, Heidi SOSIK, and Julie Huber for joining us tonight to talk about microscopic life in the ocean and its connections to marine food webs and global climate. Thank you also to my WHOI colleagues who've been working very hard behind the scenes to make this event and all our events possible. And to everyone who joined us on Zoom, Facebook, YouTube, X, and LinkedIn, thank you very much. Tonight's event was the 4th episode of our 6th season of Ocean Encounters events. We'll be taking a little break over the summer, but our next Ocean Encounters is already scheduled for September 18th. We'll be talking about the Gulf Stream and other ocean currents, so please watch our website, whoi.edu, for more details about that. In the meantime, you might want to take a look at the very cool merchandise at WHOI's online store, shop.whoi.edu. You get a 15% event discount for joining us tonight. Just type in the discount code MIGHTY. If you enjoyed tonight's Ocean Encounters program, you can be part of WHOI's solution-based approach to ocean science. If you're already a WHOI member, thank you very much. If you're not, please consider joining. You'll be providing crucial support for our outreach and our science, and you'll get some great benefits in return. Memberships start at just $40 a year. The average gift is $120, but any amount helps. A great time to make that gift is actually coming up on June 8th. That's World Ocean Day, and it's also WHOI's Day of Giving. Your support will make a difference. On behalf of Woods Hole Oceanographic Institution, thank you and good night.
Out where the seas are the deepest and the mysteries the greatest lies our future. The ocean is our last unexplored, ungoverned frontier. The life support system for our planet, inextricably linked to our climate and weather, and to the lives and livelihoods of countless people around the globe. But even against the ocean's vastness, humankind can be a formidable force. What happens next demands action that is rooted in scientific understanding and unvarnished truth, because our world stands at a fork in the road. In one direction, we watch the ocean being catastrophically altered beyond its ability to sustain us. In the other, understanding outraces exploitation, and we help steward and protect this most precious shared resource. What will be the legacy of the 21st century? Together, we can unleash the world's most creative minds, passionately dedicated to ocean science, engineering, education, and policy. Together, we can provide the expertise needed to know what works and a trusted voice to present the facts as we uncover them. Together, we can shape the future. We can inform governments, businesses, and conservationists. We can generate new knowledge in service of society. We can be the catalyst for change. It is more than our responsibility. It is the defining moment of our generation. We are explorers, students, and stewards of the ocean, and this is our time.